Okay, so the first one, cell metabolism, and by definition, I mean cell metabolism is how you break down molecules and use them for energy. That occurs in your cells. What organelle in your cell is involved in making energy? Mitochondria. Yeah. So that's where most of this takes place. Some of it takes place in the cytoplasm. And this figure in the background here, if you take biochemistry, you have to know all these pieces. You have to know all the enzymes and all the different things. You have to draw them, little guys. We aren't going to go in that much detail. We're just going to stick to the basics, but it's still going to seem probably pretty confusing. But we'll break it down, break it down into smaller pieces so it's easier to digest. Before we go into the specific processes, we're going to talk generally about the laws of physics that involve energy, which is thermodynamics. Chemistry and physics both have their fundamental origins in thermodynamics. And as we talked about before, living things or biology, the study of living things, is also, you have to integrate laws of physics and chemistry because that's what living things are made of. That's how they're constrained. So these are basics. Who here has taken physics in high school? Physical science? Okay, then you probably went over this. You have different types of motion. First one is kinetic energy. Sorry, different types of energy. Kinetic energy is energy in motion. So that's when you're actually using energy. That would be like when you are walking or riding a bike or falling down the stairs. All those things, you are using energy. Potential energy is stored energy. So that's when it's not being used. So, for example, I have a cart on top of a hill. When it is stationary, it has potential energy because of gravity. When it's moving down the hill, it is in motion, so it's kinetic energy. Once it gets to the bottom, it no longer has potential energy or kinetic energy. Okay. There are lots of forms that energy can take, so including, electricity, electrical energy, which powers our phones and all our appliances and things. Heat, sound, radioactivity, light, all those are forms of energy. But the way, and the easiest way we measure energy is through temperature or a change in temperature or heat. So for example, a calorie is a measurement of how much energy it requires to heat up 1 gram of water 1 degree Celsius. Okay, what is measured in calories? Where do you see calories? In food, right? So food, the amount of energy stored in the chemical bonds in the food you eat is also measured in calories. Anyone have some food on them? Yeah. With the nutrition label? Okay, what do you got? Red Hot. Red Hot. Yeah. How many calories per serving are 60. How many serving? 18. So if you eat 18 Red Hots, you get 60 calories. Go ahead. And that's actually a misnomer because it's actually kilocalories. So you have 60,000 kilocalories. So you could, with the energy in those Red Hots, you could heat up 6,000 grams of water 1 degree. Yeah. Okay. All right. Where does all this energy come from? The source of all our energy is what? The sun, yeah. And we can't really capture it ourselves. So how is the energy captured? I mean, we can indirectly. We can a little bit. You lay out in the sun, you can absorb the heat, but that doesn't really store the energy. So what does capture the energy from the sun on Earth? What's that? Yeah, plants. What can plants do with light? Photosynthesis, yeah. Photosynthesis takes carbon dioxide out of the air and converts it into sugar. But it needs sunlight. And how is it stored in living organisms? What stores energy in living things? What are the, how do you store energy? What's that? Fat, yeah. What is fat? Stored energy. Gross. It is molecules that are built, so it's stored within the bonds within living things. So the macromolecules that we talked about, protein, lipids, and carbohydrates, that is stored energy. Do you have a question?
I was thinking, isn't mayonnaise 98% fat?
I'm not sure, but yeah, it's...
So how do they...
It's like made of eggs.
It's made of egg whites. All right, so how do you use energy in living organisms? If it's stored in fat, how do you use that fat? You eat the fat, and that's how you absorb it into your body, but then how do you move? Yeah, so you break down those molecules, and when you break down those molecules, they give off heat and energy. So the chemical bonds are what stores the energy. It has potential energy, and when we go through chemical reactions, it releases energy, and we can use that energy to do things like move or think or whatever your body does. Oxidation reduction reactions or redox reactions are ways that we build molecules, store energy, and use molecules for energy. In an oxidation, an atom or a molecule loses an electron. And then in a reduction, an atom or a molecule gains an electron. So whenever in an oxidation-reduction reaction, you have one that's oxidized and one that's reduced. The one that's reduced gains an electron. The one that's oxidized loses an electron. The oxidized form of a molecule is the one that has the higher energy because it has the reduced, the one that has the electron has more energy. Okay, so the reduced form of the molecule is the one with more potential energy. All right, so the laws of thermodynamics, these are things by which energy is constrained. You can't break the laws of thermodynamics. The first law of thermodynamics is don't talk about thermodynamics. That's a joke. But Fight Club? Okay. All right. The real law is energy cannot be created or destroyed. It can only be changed from one form to another. So the amount of energy in the universe is finite. There's only so much. You can't make it go away. You can't make it out of nothing. It's there. But you can convert it from one form to another. So what were some of the forms we mentioned before that you can convert things to and from? Okay, heat, is one form. So how does electricity, how does it get through the wall socket? How does it get there? What is that wall socket connected to?
Wires.
Wires. Okay, where do the wires go? What's that? To the power plant. Okay, what's happening at the power plant?
There's little mice.
Little mice running on treadmills. Close.
They have their generators.
The generators. What are the generators doing? What's that?
They're
Turning?
yeah, having power.
Okay, where does the power come from?
Earth.
Probably steam.
No,
Steam?
coal.
Okay, the earth.
Sun.
The sun? It depends on the power plant, right? In Alabama, it's probably burning coal. And the coal is boiling water and turning steam, and the steam pushes these turbines. And it has these big magnets, and then the magnets create a current, and then the current goes in the wire, and then the wire goes through the outlet, and you plug it in, and your phone is charged. Okay, but it's converting it from a bunch of different forms. You're starting actually with heat. You're burning coal, and then you're making water vapor create pressure, and that pressure is going to turn these turbines, which has magnets in there. When those revolve, it converts that mechanical energy into electrical energy. Okay, so it doesn't appear out of thin air just because you plug it in. It has to come from somewhere. Right? Now, if the power plant was a solar power plant, it'd be converting sunlight into energy. Or if it was, what other types? Nuclear power. All these different forms of energy are just converting one energy into electrical energy. Okay, and I mentioned this. Total amount of energy in the universe is constant. Doesn't change. But, however, when you convert energy from one form to another, some of the energy is lost as heat. So no transfer of energy is 100% efficient. This is all the first law, yeah. It kind of blends into the second law, but yeah. So when do you feel energy being lost as heat in your daily lives? When you sweat, yeah, when you work out. So your body gets hot because you're converting the molecules in your body into energy, but then some of it is lost as heat. Okay, the second law of thermodynamics is called entropy, or the basic fundamental principle is entropy. And it is that the random order of matter is continually increasing. So things are going from an organized state to a more random state. Naturally, things are going to break down and break apart and become more random. So in other words, entropy is constantly increasing. So an example of this is your room. So after a week, after cleaning your room, what does it usually look like? A mess, right? That's mine does. And that just occurs through the natural events of going on in your life. You place something here, something there, something there. In order to get it to look clean again, what do you have to do? You have to put energy into it, right? So naturally, things will become disorganized and disordered. In order to organize them again, you have to put energy into it. All right. Free energy is a term abbreviated by the capital letter G, and this is the energy that's available for work. So because no energy transfer is 100% efficient, and some of it is lost as heat, not all of it is available for work. And so the equation we have for this, free energy equals the enthalpy minus the temperature times the entropy. So enthalpy is the energy within the bonds of a molecule. So that's basically the total potential energy. And then you have to subtract the entropy, or the energy that's not available, and that is also dependent on the temperature. Okay, in a reaction, did everyone get those? So the examples I have here is someone trying to start a fire by rubbing a stick against some wood. Here we're taking advantage of we're taking advantage of friction, which is caused by the spinning of the dowel on the wood. But as you do so, you are breaking down chemical bonds in your muscle tissue to gain energy. And then part of that is being released as heat. So as you are exercising, not all of that is converted into mechanical energy. Some of it is lost as heat. And the same thing happens to Homer when he tries to exercise. Gets hot and sweaty. All right, so you can, in a reaction, it can go one of two ways. You can gain free energy, or you can lose free energy. An endorgonic reaction. Well, let's start with exergonic. So an exergonic reaction, the products have less free energy than the reactants. Okay, so that is this one here. So this graph is marking 0 here is the amount of free energy. Okay, at the beginning. After you go through a reaction and you have products, let's say the reaction is one that we talked about before. What was the hydrogen peroxide one? H2O2. And what did it break down into? We did this one in lab. What's that? Water and oxygen. Yeah. Let's see. Just put a 2 here. Okay, and then what was the enzyme involved that made this happen? Catalase. Good. Okay, so what happens is these are the products. Sorry, these are the products, and these are the reactants. And when hydrogen peroxide breaks down, it gives off energy. So the products have less energy than the reactants. So that's what's going on here. You have reactants, they go through a chemical reaction, the products lose energy. So you could also put plus energy. And so that's an exergonic reaction. And it's spontaneous. So it happens without having to put energy into it. All you had to do was put the catalase on the hydrogen peroxide and it happened. Okay, the opposite is an endergonic reaction, and that you have to put energy into the system. So if we wanted to combine hydrogen peroxide and water and oxygen and convert it into hydrogen peroxide, it would require energy. And our products would have more energy than our reactants. So you would gain free energy, and that would be endergonic. Okay, but these are simplified, these graphs here. It's not actually quite that simple. You do have this part in the graph here where there is some requirement of energy for the reactant to take place. And that's called the activation energy. In exergonic reactions, the rate of the reaction, so how fast it goes, depends on the activation energy. And you can increase the rate of the reaction by increasing energy or decreasing the activation energy. Okay, so the first graph we looked at just had the reactants and the products. Okay, but this one includes an amount of energy called the activation energy. Okay, catalysts lower the activation energy in a reaction. Okay, they don't change the products or the reactants, but they don't change the reaction itself. And so, so the amount of free energy required for the reaction to take place is less. And if that happens, what happens to the rate of the reaction? It goes, yeah, it speeds up. It's going to go faster. So you're going to get more product in an amount of time than you would without it. So the big thing catalysts do, and this is a good test question, is what do catalysts do to a reaction? And you would say it speeds it up. And how does it speed it up? By lowering the activation energy. Good. Okay, so now we're going to go into some of these molecules used within the cell required for energy. The first one is ATP, adenosine triphosphate. And this is like the currency. This is like money. This is what cells use to get things going, for things to work. And it doesn't store very well. This isn't how we store energy, but this is how like your muscles work by using ATP, or how you send signals from your brain to other parts of your body. It all occurs through the use of ATP. Okay, there's 3 parts to it. It has ribose, which is a sugar. We saw ribose when we talked about RNA a little bit. Then it has adenine, which is a base, and it has phosphate molecules. Okay, ATP, triphosphate, it has 3 phosphate molecules. But the bonds between them are unstable, and they also hold potential energy. When one breaks off, it gives off energy, and ATP is then converted into ADP. So adenine diphosphate only has 2, because it lost one when it broke off. If both of them break off, then you have adenine monophosphate, which only has 1. Okay. These bonds are what hold the potential energy, which can be converted into kinetic energy, which will be used in the cells for whatever metabolic processes are needed. Okay, we talked about this a little bit before about the different types of reactions. Hydrolysis of ATP releases energy. So hydrolysis requires water. You add water, and then you can remove one of the phosphate molecules, and that also releases energy. Mentioned this before, ATP doesn't store very well because the bonds are unstable. So if you can't store energy in ATP, what molecules are used to store energy? I already mentioned these. Greg? Fat is one of them. So lipids. What else has energy in it? You eat an apple. What's in there? Sugar, so carbs. Yeah, some vitamins in there too, but those aren't used for energy. So yeah, your macromolecules, proteins, carbs, and lipids. Mostly lipids is what we use to store energy. So enzymes are catalysts. So if an enzyme is a catalyst, what does it do to the activation energy of a reaction? Yeah, it speeds up the reaction, it lowers the activation energy. And it does this by binding to a molecule and somehow changing the shape so it's in a favorable position for a reaction. Lowering the activation energy will increase the rate of the reaction. Now, it's important to note the enzyme does not change. Before and after, it still retains the same shape. And they're usually made of protein. Sometimes they're made of nucleic acids. But when we talk about enzymes, we're usually talking about a protein. Okay, so we're going to talk a little bit about the shape or morphology of an enzyme. Enzymes have a specific site where the molecule will bind. The molecule that is going through the reaction is called the substrate. We learned this in lab the other day, right? When they bind together, it's called the enzyme substrate complex. So there's picture 1 right there. Our little yellow piece is our substrate, and the big purple one is the enzyme. The enzyme will apply stress to the bonds within the substrate, which changes the shape of it somehow, which lowers what? Activation energy, yeah. Okay, so you guys catching on to the theme here? You guys get all that? Lowers activation energy, which speeds up the rate of the reaction. Here's an example from your book. You have sucrose, which is made of glucose and fructose bound together. When it binds to an enzyme, that enzyme causes stress on the bonds and it causes them to break through a hydrolysis, so it requires the water molecule. Once they break, they are released, and now you have a glucose and a fructose, and the enzyme is back to its original shape. Where they bind is called the active site. When they are bound together, what is it called? Enzyme substrate complex. Yes. Okay, this is a good figure to put on an exam. I could, blank these out and ask you about the processes. What? You don't have to? Okay. You don't want me to tell you what I'm going to put on the exam? All right, so lowering the activation energy or using an enzyme will cause the rate of the reaction to change. But there are other factors as well that will change the shape. Will change the rate of the reaction. Temperature is one of them. Concentration of the reactants, products, and enzyme will change. And this was one of the questions from the lab as well. What was the third one? pH, yeah. So there is usually a spectrum of pH or temperatures or concentrations by which a reaction will take place. And then there's usually an optimum by which more of the reaction will take place at this pH of 2.5 than there will at this pH of 4. In this example, we have pepsin, which is found in your stomach. And the pH of your stomach is, guess what? Not 7. It's not 6.
4.
4, getting closer. It's around 2, 3, yeah.
What?
The pepsin is in your stomach, so it works well at between 2 and 3. Okay? Where do you think you'd find trypsin? Yeah, it's in your small intestine or duodenum and released by the pancreas. And there the pH is closer to 7. So it's going to work better there. If you put trypsin in the stomach, it's not going to work very well because it's too acidic. Another one is salinity. So how salty or how many ions are in a solution will also affect the rate of the reaction. So other things can also bind to an enzyme. This may affect the effectiveness of your enzyme, including an inhibitor, which will decrease its ability to catalyze a reaction. And you can have a competitive inhibitor, which will bind to the same site as the substrate. So this is a competitive inhibitor here because that's where the substrate would bind, but it can't because the inhibitor's there. Or you can have a non-competitive inhibitor, which will go to a different site and will somehow change that active site so it can no longer bind to the substrate. Still inhibits it, but it just binds to a different site. Okay, an allosteric enzyme is an enzyme that has an inactive form. Okay, an allosteric inhibitor then binds to a molecule at an allosteric site and decreases the enzyme activity. So this enzyme here, is that a competitive or non-competitive inhibitor? Why would you say competitive?
Compared to which one?
These are both the same. So this is our enzyme, this is the substrate, and this is the inhibitor. Is it binding to where the substrate binds or at a different site? A different site, so it's non-competitive. Okay, and when it binds there, this one's actually, that's the activator, but on this side, when it binds there, it changes the shape of where the substrate can bind, and so it can't bind it. So there are two types. So this is the allosteric activator. When it binds to the allosteric site, it enables the enzyme to bind to the substrate. And this one's an allosteric inhibitor. When it binds, it inhibits the ability to bind to the substrate. So this is the same enzyme, but these molecules are different. Because when this one binds, it changes the shape so it can bind to the substrate. When this one binds, it can't bind to the substrate. That make sense? Yeah.
When it changes it, is there anything else that can bind to it? Like anything just
Maybe. I mean, it just depends on what it is and what's floating around in the cell. But generally, no, they're pretty specific to a certain substrate.
So,
Allosteric.
What is that? Because how come it was, for instance, like the last time it was just an inhibitor, but now it's an
So allosteric is referring to enzymes that have an inactive form. So this one has an inactive form. When it binds to the inhibitor, it's inactive. Okay, this one has an inactive form when it's not bound to the allosteric activator. Okay, but this slide is referring to not if it has an inactive or active form, but whether it competes at the active site or at another site. All right, so we talked about enzymes. We talked about some thermodynamics, and now we're going to get into metabolism, which is the total number of chemical reactions that are occurring in an organism. You can have an anabolic reaction. That's where a reaction that is used to build molecules. So you heard the term anabolic steroids. Baseball players take anabolic steroids. What does that do to a person? Makes them build muscle. So you're building molecules, yeah. It's an anabolic. It creates anabolic reactions which increase the muscle mass. Okay, catabolic reactions are the opposite. They are breaking things down. And they release energy. The biochemical pathway then is the sequence of reactions required to make an anabolic reaction or to make a molecule or to break it down. It usually happens with a bunch of different enzymes, and from one step to the next, the product of one is usually the reactant of the next step. So here we have 4 different enzymes embedded within the lipid bilayer. Your initial substance goes into the enzyme and it creates a different reactant or product. That product becomes now able to be used by the next enzyme, and it's changed, and so on and so forth. So it's like a chain reaction each time it's being changed. That is a biochemical pathway. And we'll go through a few biochemical pathways. One thing that your body needs to do, or an organism needs to do, is to figure out how to stop a reaction once it's started. So if you're going to make glucose molecules, at some point your body needs to know when to stop making a glucose molecule. And it does that by using feedback mechanisms. Feedback inhibition is one way in which the product also becomes an allosteric inhibitor to the enzyme in the pathway. So let's say in this product we're making, or in this biochemical pathway we're making glucose, or we need glucose. And so our enzymes will take our substrate and convert it into glucose. Well, in a allosteric inhibitor, our glucose will then bind to our enzyme, and that will cause our initial reaction to stop. So that when we have too much or enough glucose, it'll prevent the biochemical pathway from making more. Keeps you from overproducing the substance. All right, so there are two kinds of respiration. There's breathing in and out, and then there's cellular respiration. We're here are talking about cellular respiration, which is converting organic molecules and oxygen into ATP, which can be used for energy. Autotrophs are organisms that can photosynthesize, can take energy from sunlight, and with carbon dioxide convert it into organic compounds. Heterotrophs are organisms that can't photosynthesize and can only receive energy by eating plants or other animals or other organisms. So which of these is an autotroph and which is a heterotroph? Yeah, the plant. The cow eats autotrophs. This guy's green, but he does not photosynthesize. Caterpillar. Okay, cellular respiration is a series of reactions or a biochemical pathway where you start with an organic molecule, and we're going to start with glucose and go through it. And then we break it down to create a proton gradient, and then that proton gradient is used to convert ADP into ATP. And redox reactions are vital for creating this. I photoshopped a Redbox logo and made it redox. So, do you remember what an oxidation reaction is? A molecule that is oxidized gains an electron. Sorry, loses an electron. And then a molecule that is reduced gains an electron. In a dehydrogenation, an electron is lost, but it's accompanied with protons. So if you lose an electron and a proton, that would be a hydrogen atom. Okay, so an example of this is NAD+, which is used in a lot of biochemical pathways. In a reduction reaction, it gains an electron and it becomes NADH. It also gains a proton and 2 electrons. In an oxidation reaction, NADH loses that proton and 2 electrons and becomes NAD+. Okay, and it just makes sense that it would gain a proton and 2 electrons. With NAD plus, it is an ion. What type of ion is it if it has a positive charge? What type of ion is it? A cation, right? There's a positive charge. How many electrons do you need to make it a neutral then?
One more than
Yeah, 1. So if you added 1 electron, it would be NAD. Okay, but then you also gain another electron and another hydrogen, or another proton, which is a hydrogen atom. So it becomes NADH. All right, the sequence of redox reactions in cellular metabolism oxidizes organic compounds. So you're taking glucose or your lipids and you oxidize them, so you're taking electrons away from them and protons. And then it also reduces NAD plus and FAD plus. FAD plus is a very similar molecule to NAD plus. But it reduces them to NADH and FADH2. Then FADH2 and NADH are oxidized, and O2 is reduced and becomes water. So this is the reason why you breathe in oxygen. It's so that when you break down glucose or other molecules, you have an acceptor for electrons, and it's converted into water. And we'll go into more detail in a minute. So again, oxygen is the final electron acceptor in aerobic respiration. In anaerobic respiration, oxygen is not the final acceptor. They use other molecules. So this happens in bacteria that live in hydrothermal vents or in geysers, or also happens in fermented things that ferment. This is material that is made out of fermented grapes, sort of cloth. I found on the internet. But the fermentation process uses something besides oxygen, so it's anaerobic. And in fermentation, the final electron acceptor is an organic molecule. All right. In aerobic respiration, the change in free energy from one molecule of glucose is 686 kilocalories. But this isn't released all at once. It's released in a bunch of steps. So it's kind of like a staircase, and each step in the process, in the biochemical pathway, releases a little bit of energy, and then that energy is captured all at the end to make ATP. This is the general equation for respiration. You guys have that written down in there? Is it on the slide? If not, write it in, because this is one that will show up on an exam. And this is the opposite of photosynthesis. In photosynthesis, you start with carbon dioxide and water, you add sunlight, and you get a sugar molecule and oxygen.
so it's the opposite of it.
Photosynthesis. All right, so I've said this. Did you guys get all that? So our cells use ATP to drive endergonic or catabolic reactions. And the change in free energy in ATP is only 7.3 kilocalories per mole. There's two mechanisms for synthesis. The first one is substrate level phosphorylation, which transfers phosphate group directly to the ADP, and this happens during glycolysis. We're going to talk about glycolysis in a second. The second one is oxidative phosphorylation, which uses an enzyme called ATP synthase, and this occurs in the mitochondria. So that word glycolysis, what does the suffix lysis mean? Remember what a lysosome does? That lys means to cut. So glycolysis is the cutting or the splitting of glucose molecules. Okay, and that's what the first step is. We're oxidizing glucose. First thing we're going to do is we're going to cut glucose and make it into pyruvate molecules. Then we're going to oxidize pyruvate. We're going to take those oxidized pyruvate molecules and send them through a series of chemical reactions called the Krebs cycle or the citric acid cycle. And that's going to convert or reduce NAD plus and FADH and FAD molecules. And then we're going to use those electrons to push protons through a proton pump, and that's going to make ATP. So these are all the steps, but we're going to go through them individually. Yeah, we still have a lot left. But we're almost done with the first lecture, right? Okay. Glycolysis. Again, you're cutting glucose molecules. And glucose is a 6-carbon molecule, and you're going to cut it into 2 3-carbon molecules called pyruvate. This occurs in the cytoplasm. So I'm going to do a little drawing with this. All I'm going to draw is, this is a cell. I'm not going to draw all the organelles. I'm just going to draw a chloroplast, not a chloroplast, a mitochondria. So out here is the cytoplasm. This is where glycolysis occurs. Okay, when you break glucose into pyruvate molecules, you get 2 ATP. So already we're getting energy. And you also reduce 2 NAD plus molecules into NADH. All right, so of these 4 molecules, which have the most potential energy? ATP, right? How about between NAD plus and NADH? (50-50). Yes, NADH. Because it has a molecule of hydrogen on there. NAD plus has a less, does not. Okay, that H, that bond there, is holding energy. And ATP has 3 phosphate molecules, ADP has 2, so it has an extra bond there. That bond has more potential energy. We can use that ATP for energy. The NADH we have to convert into something else, which we'll do later. So for glycolysis to continue, NADH, you need NAD+. So you have to convert that NADH back to NAD+ if you want to keep on doing glycolysis. So you can do this by using your NADH in aerobic respiration, which gives you a lot of ATP, or you can go through fermentation. So when oxygen is not available, or when you're working too hard, When you do very strenuous exercise, you don't have enough oxygen in your muscle cells or in your cells. So your body will switch from aerobic respiration to fermentation. Well, it won't switch. It will do that in addition to aerobic respiration. Okay? And this doesn't give you as much energy, but it gives you quicker energy. So when you're like lifting weights or you're doing these short bursts of energy or you're sprinting across the room, you're using aerobic respiration and fermentation. But if you do have oxygen, you're going to take that pyruvate and you're going to oxidize it. This occurs in the mitochondria. So glycolysis is that, and then pyruvate oxidation. I'm just going to put PO. It occurs at the plasma membrane in the mitochondria in eukaryotes. In prokaryotes, it occurs at the mitochondrial membrane in eukaryotes and at the plasma membrane in prokaryotes. When you oxidize pyruvate, it turns into acetyl-CoA. You don't have to know this. But when you do that, it emits carbon dioxide, which is a waste molecule. We can't use that for anything. Another NADH molecule, and then your acetyl-CoA. All right, that's it for that one. We're halfway done. I don't know if we're halfway done. Do you guys want to take a break?
Yes.
Okay, let's take a 5-minute break. Okay, so after you have this acetyl-CoA from pyruvate through pyruvate oxidation, you then go through the Krebs cycle, which occurs in this matrix within the intermembrane. And that oxidizes the acetyl group from pyruvate and is a really long, complicated step, lots of steps. It eventually converts acetyl-CoA into oxoalloacetate. Okay, right? And then you get a new acetyl-CoA that combines with oxoalloacetate and forms a 6-carbon molecule called citrate. So sometimes it's called the citric acid cycle. Then that's rearranged and decarboxylized. So it loses hydrogens, it loses electrons, and then it regenerates oxoalloacetate. But the important thing to note in here is not all these names. That's fine if you don't know those. But what we're making is FADH2 molecules and NADH molecules. And we do get some ATP, and then the byproduct is carbon dioxide. So that's why we breathe out carbon dioxide. It's because when we go through the Krebs cycle, it's a byproduct that we can't use anymore. For each acetyl-CoA, you get 2 molecules of CO2. Not worth anything. We get 3 NADH molecules, and we get 1 FADH2 molecules. And we get 1 ATP. So, what was I going to say? But we're going to then use these NADH and FADH2 molecules. Oh, I was going to say, we have 2 pyruvate molecules or 2 acetyl-CoA molecules per glucose. So this happens twice, and you have 2 ATPs, 2 FADH2s, and 3 NADHs per molecule of glucose. And so that's why everything's doubled on this slide. So for every molecule of glucose, once it's gone through the Krebs cycle, you have 6 carbon dioxides, 4 ATPs. We had some of the carbon dioxides from pyruvate oxidation. 10 NADHs and 2 FADH2s. And now we have all molecules necessary, these guys, FADH, NADH, to convert, to make some ATP. You get all those? Okay, and this happens through what's called the electron transport chain. Okay, this is a bunch of integral plasma membrane proteins on the inner mitochondrial membrane. So they're embedded in here. Okay, and what happens is you take those NADH molecules and FADH2 molecules that we had before, and you oxidize them. You take away their hydrogens, their electrons, and their protons, and you spit the hydrogens on the inside here. And so now you have more hydrogens on the inside than you do on the outside. So which way are the hydrogens going to want to go? On the outside. Why? What's that process called? Not osmosis, diffusion. Yeah, and facilitated diffusion, because it's actually going to go through a certain molecule, and that is ATP synthase. And when it goes through that molecule, or through that enzyme embedded within the layer, it's going to make ATP. Okay? This proton pump is ATP synthase, so that's the same thing. So for every molecule, so chemiosmosis is this process of putting all these protons on one side, and then as they pass through ATP synthase, they convert ADP into ATP. Okay, it's chemiosmosis because our chemical here are these hydrogen ions, and they're flowing through a semi-permeable membrane.
it semi-permeable?
Yeah. Yes. So the inner mitochondrial membrane, just like the plasma membrane, is semi-permeable. Okay, so this is the overall slide. This is what we just drew, basically. We started with glycolysis, and then we had pyruvate oxidation, which created acetyl-CoA. That was used in the Krebs cycle. Krebs cycle makes FADH and NADH, which is then used in the electron transport chain, which creates a proton gradient. And when the protons go back into the other side, they create ATP. So I had this drawn backwards. Hydrogens go out here. And then go inside.
So which part on there is the TPS?
Chemiosmosis is these hydrogens going through the proton pump. Yeah. All right. Is that clear? You guys all understand that perfectly? Ready for a quiz?
Yeah.
All right. So the theoretical energy yield is 38 ATP for glucose in bacteria, or 36 in eukaryotes. So that's how much we get out of one molecule of glucose. But actually, we get a little bit less than that. We only get 30 because some of the transporting molecules from one place to another costs energy, and the membrane isn't sealed completely, so some of those protons get through and they aren't converted into energy. Okay, I mentioned this before, but sometimes you can have respiration without oxygen, and an anaerobic or an inorganic molecule will be used as the final electron acceptor. Some prokaryotes use sulfur, nitrate, and carbon dioxide. An example are some methanogens and sulfur bacteria, and these are found again. And you ever gone to a hot springs that stinks like sulfur? You can sometimes find bacteria there that are using sulfur compounds rather than oxygen to go through respiration. Fermentation happens through all organisms are capable of fermenting. We do lactic acid fermentation. Yeast does alcoholic fermentation, and so that's how alcoholic beverages are made, by adding yeast to your grape juice. They ferment the juice and create alcohol. So in fermentation, the organic molecule is the final electron acceptor. I just basically said this. So yeast does ethanol, which is alcohol. And the byproducts are ethanol, NAD+, and CO2. And then our muscle cells will go through lactic acid fermentation. And the byproduct is lactic acid instead of ethanol. Okay, so as I talked about before, not all molecules are made out of glucose. What molecules are made out of glucose or made out of sugars? DNA has a sugar in it, yeah. What other molecules? The monomers of which are sugar molecules. Corn? What?
I think
Carbs, yeah. Carbohydrates are made out of strings of sugar molecules. But proteins and fatty acids are not made out of glucose. They have different molecules in there. But they can also be used and broken down into energy. And in order to do that, though, they have to be converted into a product or reactant in the Krebs cycle, or it can be converted into glucose molecules. So in amino acids, they have to undergo deamination. So they have to remove their amino group, which is the nitrogen part. And this happens, so when you eat meat and you use it for energy, you take off the amino acid group, and then your body converts it into urea because the amino group is very reactive. And so that's why you pee, or that's one of the major components of pee is urea. That's also why it's called urine. Fats are broken down into fatty acids and glycerol, bless you, through a process called beta oxidation. Okay, and it's basically just breaking that fatty acid into a bunch of different chunks, and each one of those chunks can then be used in the Krebs cycle. And this yields much more energy than a glucose molecule. So that's why we store energy in fat molecules or in lipids, because they can store more energy than in glucose molecules. One 6-carbon fatty acid yields 20% more energy than our 6-carbon glucose. Okay, so we'll do this last drawing, and then we'll be done. I'll just record the photosynthesis stuff. Okay, so one way we can get energy is through glucose. Okay, what's the process that it has to go through, though, is glycolysis and pyruvate oxidation. Okay, if we want to use amino acids, they have to go through deamination, eternal deamination. If we want to use fatty acids, let's see. If we want to use lipids or fats, we'll just say lipids. They have to go through beta oxidation. So that weird-looking B on there is not a B. It's the Greek letter beta. And so those are of this figure, what I want you to know are these basics. Okay? The Krebs cycle, in the end, you get carbon dioxide, which is your waste. You get water. When you go through deamination, you're taking off your amino group, which is NH3. And that's it. So if you know this, you don't have to know everything on here. All right, we're gonna do I'll do photosynthesis. I'll record it and post it to YouTube, and you guys can fill it in on your own time. Next Thursday, you're being tested on these 2 chapters, though. So start studying today, I heard some people say they just started studying last night. That might be a little late, so start studying today and get this stuff in your head, and then it won't be such a big deal last second. Okay, go ahead and leave.
Hate when that happens.